Okay, so this is our first unit, and our first lecture is going to talk about macromolecules and metabolism and just get introducing you to enzymes. So first, just to give you some information, there's a good chance I'll have to modify my Monday office hours, so I will let you know about that next week as soon as I know that. And because this is an out-of-class lecture, you are going to have an assignment to complete to make sure that you actually watch the lecture, to make sure that you actually get the information that you need for the class. And just a reminder that, if you have any questions about this material after you watch the lecture and actually after hopefully bad winter weather has passed, please come and see me or email me so you can make sure that you are all set and good. So what we're going to do is the first part of this lecture is going to focus on macromolecules. And these are the four major organic molecules that are necessary for life to occur. And so this includes proteins, nucleic acids, carbohydrates, and lipids. And these are doing a ton of work in the cell in terms of providing structure, energy, and of course the genetic information. So what we're going to do is we're going to go through each of these. We're going to talk about their structure and their function. You are going to have to be able to visually recognize them and also recognize the bonds that form. So a lot of these are what we call polymers. So these are molecules that are composed of many different smaller subunits, which are called monomers. And these can be repeated similar subunits in some of these molecules. So, they're very similar in their basic structure, like the bases, the nucleotides that we see in like DNA and RNA. But there are some slight differences. So like what base is attached or what amino acids being added. So we're going to talk about each of these molecules and how they are put together and of course their function. So the real workhorses of the cell are proteins. And so proteins are doing pretty much everything in the cell. There are the enzymes that are going to carry out cellular reactions that are necessary to maintain life. We're going to talk about those later. There are receptors in cell membranes that allow for selective signaling through the cell membrane where molecules on the inside and outside are talking to each other. They're transporting things inside the cell or across the cell membrane. And of course, they are providing structure and support. So we should know from 148 already that proteins are composed of these amino acid subunits, and these are linked together by peptide bonds. So remember that peptide bonds occur between the carboxyl group of one amino acid and the amino group of the other. And so this should already look familiar to you, but we consider the amino acids to be the monomers that form the polymer protein. And just one thing that I want to note, because it's really important to remember that for proteins to function, they have to be folded correctly. So remember, we've talked about these different levels of protein structure. We have primary, secondary, and tertiary. This is once again something you should remember from 148. But that tertiary structure has to be folded into that 3D structure in order to function properly. And so, we're going to talk more about, like, for example, enzymes. If they're at the wrong pH or wrong temperature, they could not work properly. And then as a result, your enzymatic reactions are not going to be carried out at all, basically. Now, nucleic acids, we know that they always they carry the genetic information. So we know this with RNA and DNA. But something you may not know is that nucleic acids are really important in serving as energy molecules inside the cell. So you can see that here on the right side, this is ATP, that stands for adenosine triphosphate. So this is a modified adenine base. And this is the primary energy currency found in the cell. We're going to talk a lot about ATP when we talk about cellular respiration and photosynthesis. And so ATP is really important in making sure that your cell can function. Another common energy currency is GTP, so guanosine triphosphate. So nucleic acids not only carry our genetic information, but they also are these energy carrier molecules. Now, when we link them together to form the RNA or DNA, they are composed of these nucleotide monomers. And so remember, the nucleotides is composed of a sugar, the nitrogenous base, and the phosphate. And so we have phosphodiester bonds that form between the 3' OH of one nucleotide and the phosphate group of the nucleotide that's being added to the chain. And once again, this should look very familiar because we did cover this in 148. The next group are carbohydrates, and these are, your sugars. And these are going to be the energy for metabolism that we use. So we're going to talk about cellular respiration. And cellular respiration is, and part of it's the breakdown of glucose, which is a sugar, to help produce ATP that your cells need to function. Now we can have simple sugars, which are called monosaccharides. So these are going to be linear or cyclic molecules. Usually they have 5 or 6 carbons. And you can see that here. So you can see things like the glucose and the fructose. Ribose, which is used in DNA, so your nucleotide and RNA, your nucleotide subunits have a monosaccharide contained within them. And so these are really simple. These are just one little structure composed of 5 or 6 atoms. Now we can link these monosaccharides together. Okay, so quick question, stop and think. Which of the following shows the correct relationship between the monomer subunit and the bond that would link them together? So take a quick look. And the answer is none of the above. So nucleic acids are linked together by phosphodiester bonds. Amino acids are linked together by peptide bonds. Monosaccharides are linked together by glycosidic bonds. So none of these are the correct pairing for these molecules. Okay, so the next group, these are not going to be polymers. These are going to be just individual little carbon molecules, but they all share something in common, that they have hydrophobic components. And so these are the lipids. And so there are 3 different types of lipids: fats, steroids, and phospholipids. And so you can see the top here is a cholesterol. That is going to be a steroid. The triglyceride you see, that's a fat. And then on the very bottom, the phospholipids. So we're going to talk about the function and structure of each one of these. So fats, you might also hear them referred to as triglycerides. These are nonpolar molecules, and the thing that will help you distinguish them visually is that they have these 3 fatty acid chains. So you can see these 3, if you look at the very bottom picture, you see those 3 like long carbon chains coming off. Those are fatty acid chains. And so those are connected together to, if you look at the very front of the triglyceride, this what's called a glycerol. And so, of course, we know that fats are used for energy storage. A lot of animals maintain fat reserves so that, when food is scarce, they are able to break the fat down to make ATP, for example. The next lipids are steroids, and this is not like, I don't want to think about like, a bodybuilder, with steroids. Steroids are actually very important to the function of your body. So one really important steroid is cholesterol, and this is a major component of the animal plasma membrane. We're going to talk about how important cholesterol is in maintaining fluidity in the cell membrane. And they're also utilized as signaling molecules. So steroids includes estrogen and testosterone. And so a lot of hormones are steroids. And so these are going to, travel throughout the body and cause things to happen. So, steroids, especially like during puberty, are going to signal changes in the body. You can denote steroids from these bulky ringed structures that you see. So you'll see multiple rings together making up the steroid. Next are the phospholipids, and we're going to talk a lot about phospholipids over the next few lectures because these are the major component of the cell membrane. And the way to denote a phospholipid is that there are two fatty acid chains at the bottom. They're attached to a glycerol, and then that's attached to a phosphate. And the way that this is structured is that it actually has a hydrophilic head, so a water-loving head, and a hydrophobic tail, which is water-fearing tail. And in animals, they're fatty acids. There are some species like Archaea where they actually have isoprenoids as their hydrocarbon chains. So take a look at this figure here and try to think to yourself if you can identify what type of lipid is shown here. So take a second. And the answer is A. So this is a fat. And remember that the way that we denote a fat is that you're going to see these three hydrocarbon chains. So we talked about these four classes of macromolecules, and we have these monomers linked together, but these linkages don't just happen on their own. If we waited for them to happen on their own, it would take a really, really long time to happen. We have we have these molecules that store energy, and so sometimes we need to harvest that energy, for example, like the fats, if we need to break those fats down and get the energy that's found in those fats. And so we use enzymatic reactions to break down molecules or put molecules together. So we're going to talk about metabolism now and then just briefly talk about enzymes. So metabolism. So these are all the chemical reactions that are happening in your cells, and they are converting one type of molecule into another or putting two types of molecules together to make a new one. And all of these processes are happening to get energy or transfer energy in order to sustain life. And so we're going to build things up or break things down, or in that process, we're converting things as well. So we're changing molecules in some way. And by doing this, we're harnessing energy. And once again, these are all the chemical reactions. And you can see this is showing you many metabolic pathways that are linked together. And so you're going to have products produced by one metabolic pathway feeding into other metabolic pathways as new substrates or molecules that are going to be linked together. So all of these work together in some way. Now, metabolism is divided into two major types, what we call catabolism and anabolism. And so we're going to talk about the difference between the two. So catabolism, think of catabolism as a catastrophe. So something's going to break. And so catabolism is breaking down molecules into smaller units. So think about this as, breaking down, a sugar into its smaller units. And when you do this, you're making ATP. So this is a lot of the times you're breaking things down to make ATP. That's catabolism. Anabolism is putting things together. So you can sort of think of like anabolism, add things together. So you're going to be putting molecules together, and then these are these smaller monomers linking them into polymers. And once again, this is going to require an input of ATP for this to happen. So catabolism, break things down, produce ATP. Anabolism, build things up, use up ATP. So if we were to break a glycosidic bond, what would this be? Would this be catabolism or anabolism? So think to yourself real quick. And the answer is catabolism, right? So catabolism, catastrophe, we're breaking things, destroying things, and so breaking a glycosidic bond would be an example of that. Now we need this to happen at a really fast rate, okay? We can't just wait for molecules to bump into each other whenever they are able to, because if we relied on that, we would be dead, okay? So these are the chemical reactions that we need to maintain life. And so for this to happen, we have biological catalysts. So, catalysts, if we think to chemistry, they speed up chemical reactions. And so we're speeding up the chemical reactions in the cell. And so these biological catalysts, we know them as enzymes. Okay, and enzymes are proteins. So you remember I talked about, we need, proteins are doing the main work of the body, and this is one of the ways that they do that. They are proteins that are helping to facilitate these chemical reactions to occur. Now, enzymes both break down and put things together. So in this particular image, you can see the substrate. So this is, you'll hear this referred to as a substrate or the reactant that's going to, in this particular case, you can see that this substrate is broken down into two separate pieces, and so that would be our product. We could have the reverse reaction where these two separate little product pieces are coming together in the enzyme and then leaving as this linked molecule. And so once again, it's not just breaking things down. Please do remember that it also puts things together. And enzymes are really important in helping chemical reactions occur because what they're doing is they are placing these molecules together, these reactant substrates, in a very precise orientation to facilitate bond formation. If you just rely on these substrate molecules to bump into one another, then they may not bump into one another in the right orientation to facilitate how the bond is formed. You have to have a certain amount of kinetic energy to either break bonds or make a new bond. And of course, you have to consider that these are molecules, there are electrons, and so there's some repulsion that comes with that. And so the enzymes are going to make sure that these molecules are coming together in just the right way so the electrons can interact with each other and form bonds. And so these substrate molecules interact in a very specific site in the enzyme, and that's called the active site. And this active site is very specific to the type of substrate that it binds. Most enzymes only catalyze one enzymatic reaction. So you can sort of think of it as a lock and key. So you have, one key that opens only one lock. With the enzyme, there's only one substrate that can bind the one active site. And so the enzymes are going to help the substrates collide in a precise orientation, and that they will enter the active site in a very precise manner and interact with one another to facilitate the bond formation. That active site is usually this sort of like little indentation in the enzyme. So enzymes are usually these globular proteins. So you can see that here. And the blue in that image is the active site. And so it's going to be this tiny like indentation or this cleft or cavity within that enzyme where these molecules can enter and interact with one another. And when they enter and interact with one another, what happens is a change in the conformation of that enzyme. This is what we call an induced fit. And basically, it's sort of the enzyme like wrapping itself around those substrates to put them in that very precise orientation that they need to facilitate bond formation. And so you can sort of see that between the top and the bottom. So you can sort of see how at the top it's kind of open, and then once those molecules come in, you can see like the blue color has shifted. We're sort of closer together. And so this induced fit, this conformational change in the enzyme, is very important to positioning those substrates together in order to form a product. Okay, so this is where we're going to stop. We're going to pick up with enzymes again next Wednesday. Thank you so much for being so understanding about me canceling class. Please make sure to complete the assignment for this so that you can get your points.